Hey everyone, Azim here. We are continuing our talk on the nervous system. We finished talking about the brain, not exactly. We're going to focus here on the cranial nerves and talk about different brain regions that relate to cranial nerves. So this is chapter 15b, cranial nerves. We'll be identifying the different cranial nerves that enter and exit the brain and talk about their functions, of course, basic functions. And it'll be important, of course, to identify the different cranial nerves that come out of the brain, the different pathways to and from the brain. If they're sensory, that's to the brain. If they're motor efferent, that's from the brain. And how to assess, how to test their proper functioning. If you've ever been to the doctor, which I hope you've at least been a couple times in your life just for basic checkups, you'll notice that they do these funny things to you. They cover your eye and make you follow their finger, or they ask you to shrug against their shoulders or shrug your shoulders against their hands. They ask you to open your mouth and stick your tongue out and blow your cheeks out and prevent your cheeks from getting pushed in. They make you close your eyes and touch something on your face. These are all tests. All these funny things they do are tests to make sure that your nervous system is working correctly. Specifically, the tests shown here are cranial nerve tests. Very simple, only takes a few seconds, tests that can effectively see whether or not your nerves are working properly. And if, for instance, you don't feel anything on your face, or you're not able to stick your tongue out, or you can't shrug against someone's hands, then they would do more investigating, because that would suggest that there's a problem. We're going to, once we go through this chapter, we'll be able to identify what different tests we can do for these different nerves. There are 12 pairs of cranial nerves, 12 pairs. Cranial, because they come off of the brain. That's why they're called cranial nerves versus spinal nerves, which we'll learn later. And nerve, if you recall, nerve is a group of axons outside of the nervous system, outside of the central nervous system. So they're in the peripheral nervous system. So the brain is the central nervous system. Nerves are axons, not full neurons, just the axons found outside of the central nervous system. They're in the peripheral nervous system, but they go to and from the brain. They're all found on this ventrolateral, really this like inferior-ish surface of the brain. So we're looking at the underside of the brain here, and we can see all the nerves coming off of the brain. I missed one. Here, 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 and yeah, I think I got it all. We'll go over all of these different nerves in this chapter. There's a lot of them. They look really small in some places, and that can get really confusing, but I'll give you tips on how to recognize where they can be found and what they do. You'll also notice that they're numbered 1 through 12 in Roman numerals. We've only gone through maybe Roman numerals through 5 because of the phalanges, 1, 2, 3, 4, 5. If you're unfamiliar with Roman numerals, it gets weird around 4 and 5. 4 is IV, 5 is V, 6 is VI, so that's just one after 6, or one after 5. So 4 is weird because it's 1 before 5. 9 is weird because it's 1 before 10. 10 is X. And then we have 11 and 12, XI and XII. So brush up on Roman numerals if that's something that is unfamiliar with you, unfamiliar to you, but you will need to know these Roman numerals. These nerves, these cranial nerves, have to pass through our skulls to different organs. We have nerves going to our eye, to our whole face, to different parts of the ear, all over our body. And so they need to pass through bone. And if you recall, we have holes in our bone, especially tons of holes in our skull, called foramina or canals or a meatus, different passageways for nerves to pass through. And summarized here are all the different passageways and the corresponding nerves that pass through those passageways. So we'll refer back to this when we go through each one. The first nerve, the first cranial nerve, is the olfactory nerve. Olfactory, that's referring to smell. So it's the most anterior of all these nerves, which makes sense because it's the one closest to the nose. And you smell with your nose, of course. It's the only cranial nerve that's not attached to the brainstem. If you take a look at this underside of the brainstem is over here, and you can see all of the nerves that I'm circling here are coming out of the brainstem. The olfactory nerve, though, is coming out of here, where I'm drawing these lines. This is an underside view of the brain, where I'm going to draw it in pink over here. This is a mid-sagittal view, which includes the brain, but also the nasal cavity. And you can see nerves going up and down. They're not that long, but those are nerves. Those are bundles of axons extending from the olfactory epithelium up to this structure that I'm going to color in blue, called the olfactory bulb. So there are neurons that start in the nasal epithelium, in the olfactory epithelium. They go through holes in the bone. They go through holes of the olfactory foramina within the cribriform plate of the, recall that this is part of the ethmoid bone. And then they reach a structure called the olfactory bulb. The olfactory bulb is this region of the brain that we haven't gone over yet. Here it is. It's this region of the brain that's on the underside of the brain, a bit anterior rostral. So, olfactory nerve carries sensory information, about what it is we smell. A molecule in the air dissolves in mucus, goes into our nasal epithelium, binds to some dendrites of neurons. So there are neurons here. Binds to the dendrites of neurons, send a signal along the axon to our brain. That's the beginning of the smell pathway, olfactory pathway. Here's another view. So same thing that we're looking at, just the nose is facing the other way. If I draw a single neuron in this pathway, let me draw a single neuron here in green. Remember, this is how I draw dendrites, this open caret. These are dendrites. We get dendrites extending to a cell body. This is a special sense. Olfaction is a special sense. If you recall, for special senses, what shape are these neurons? These neurons are bipolar shaped. So there's our cell body. There's our axon. There's our axon terminal. This is a bipolar sensory neuron in the peripheral nervous system. Dendrites, cell body are outside of the central nervous system. The axon terminal ends in the central nervous system. It's in that olfactory bulb. That axon is part of cranial nerve 1. All of these axons that are going up through the bone, that collectively is cranial nerve 1. So these olfactory neurons send a signal up to the olfactory bulb. This is the olfactory bulb. It's this bulbous shaped structure. Looks like rabbit ears on the underside of the brain. And then that neuron will communicate with another neuron. So I'm going to draw another neuron here in the olfactory bulb. It's multipolar because now we're in the central nervous system. And it's going to send a signal along an axon to another brain region. Its axon, since it's in the central nervous system, it's no longer called a nerve. The group of axons in the central nervous system, this is called a tract. The olfactory tract is this group of axons passing along in the central nervous system. And this second neuron ends, so its axon terminal ends at the olfactory cortex. The olfactory cortex is a brain region that's kind of on the border, as you can see, it's on the border of the frontal and temporal lobes. This is the main region for processing smell. Olfactory cortex on the border of the frontal and temporal lobes is the main area for processing basic smells. From here, if we want to think more cognitively about the smell, like try to identify what it is, and think about other properties of the smell, we can then send a signal with another neuron, another multipolar neuron, and send that to somewhere else in the cerebral cortex. I'm being intentionally vague here because it can get really complex. So another multipolar interneuron. But generally, if you want to think more about the smell, send it to somewhere else in the cerebral cortex, probably through an association, through association fibers to other parts of the cortex. If we want to think about that smell in context of what does it remind us of? Do we have a memory of that? Or does it make us feel a way? Like some smells make you feel really happy. Some smells make you feel really scared because you associate something, say, with your childhood or some really weird traumatic memory. There's a group of brain regions that are important for emotion and memory. That group of brain regions is known as the limbic system. And so we could have another neuron send information to some place in the limbic system. I've mentioned the limbic system. I showed some parts in the last video. Let's review what those parts are. Limbic system is made up of 5-ish places here that I've listed here. That's generally important for regulating, processing emotion and memory. This first one, called the cingulate gyrus, is a brain region that immediately surrounds the corpus callosum. Very generally important for emotion. So you could be sending information here. The hippocampus is a brain region a little more lateral and curved, like a seahorse. That's what hippocampus means. Hippocampus means seahorse. The hippocampus is involved in learning and memory. It's affected in conditions like Alzheimer's disease. It is affected in other traumatic injuries, like I've mentioned this, if you've seen 50 First States or Memento, those are damage, that's damage to the hippocampus. We've seen conditions like this before. The fornix is a white matter tract that extends out of the hippocampus, so it's axons, and that fornix ends at the mammillary body, and the mammillary body relays information from the hippocampus to the thalamus. And remember, the mammillary body is on the underside of the brain, that inferior ventral part of the brain. The amygdala is this rounded structure region that's also pretty lateral towards the ends there of the hippocampus. And the amygdala is involved in fear learning, fear and memory. If you've ever seen Free Solo, that documentary about that guy who climbs where was it? Yosemite, I forget the name of it, El Capitan. He climbs really high places without any support, without any safety gear, just free climbing, free solo. They did a a brain scan of him and to look at his activity, and they found really low levels of activation in his amygdala. That suggests that he has very low baseline level of fear. In most people, you get afraid of really high heights or anything that might threaten your safety, and those things don't really frighten him. So in most people, the amygdala is important for regulating fear when it's necessary, when it's associated with a threat to your life, to your being. So all these parts are part of the limbic system, and we could associate smell. We could send smell information to any parts of these if we associate that smell with fear or with just something from our childhood or with a general emotion. And it doesn't just have to be smell either. We could do this with vision. We could do this with hearing, other senses. But there's a million different pathways that we could go over. We're only going to go over some of them. Second cranial nerve. Second cranial nerve is the optic nerve. Optic, as this name is relating to, it's relating to the eyes. There are other cranial nerves that relate to the eyes, so got to be careful. But in this case, optic nerve is a sensory nerve. It's important for vision. So first nerve is sensory for smell. Second nerve is also sensory. It's for vision, the optic nerve. This starts at a place called the retina. The retina is the back layer of the eye. It's a back inner layer. Whoopsies. It's the back inner layer of your eye. So if we take a look at the eye over here, for instance, it would be this layer that I'm coloring in blue. That would be the retina. So not the one that's most superficial, it's the deeper inner layer. The nerve starts at the retina, and goes to the brain. The hole that it goes through is called the optic canal. The hole that it goes through is the optic canal. The optic canal, if you recall, is part of the sphenoid bone. It's kind of hiding where that sella turcica is. There's one on either side. These optic nerves, they send information to the brain, and the axons continue, but there is this weird crossing point. If you look at over here, there's this area where the axons cross, because remember, nerves are axons, and here we have a crossing of axons. Some axons will just stay on one side, but then sometimes you'll get axons that will cross over from one side to the other. So you can see I've started an axon from this left side. We're looking at the inferior of the brain right now. From this left side over to the right side. We get a crossing over sometimes. The term for this region is the optic chiasm. The optic chiasm is a region where we sometimes have a crossing over of visual information. When do axons cross? Let me erase my markers here. Sensory information about vision, they cross over depending on visual field. If this is the right eye and this is the left eye, if I see something in my right visual field, If I see something in my right visual field with my right eye, if I see something in my right visual field with my right eye, it's going to be processed by my left brain. Right visual field is processed in the opposite side. It's contralateral processing of visual field. Let me write that down. Contralateral processing of what you see in your visual field. If I see something in my right visual field, it's going to get processed in my left visual cortex. How does it get there? We go from right visual field in this part of the right eye. It will cross over at the chiasm and continue to the left side. And then from this left side, it'll continue on to the visual cortex. Compare that with, say, I see something in my left visual field with my right eye. If I see something in my left visual field with my right eye, so it's hitting this part of my eye, the lateral part of my eye, it's going to stay on the same side. It's going to stay ipsilateral. So at the chiasm, there was no crossing. I know that's a little confusing, but I'll do this one more time. If you see something in your left visual field, it's going to be processed by your right brain. Left visual field gets processed in your right brain. Left visual field, this will cross over, gets processed in your right brain. Right visual field, oops. Right visual field gets processed in your left brain. Right visual field gets processed in your left brain. It just depends on which side of the eye it hits. This portion that I'm coloring in blue, these are the optic nerves. These are the bundles of axons that carry information from the eye to the brain. Once we're in the brain, once we're past the chiasm, we're in the brain. And once we're in the brain, we don't call bundles of axons nerves anymore. We call them tracts. So what's funny is that our axons didn't stop. They didn't stop. We didn't like end at another neuron. Tracts are just a continuation of those same axons, but we don't call them nerves anymore. We call them tracts because they're now in the central nervous system. The tracts end finally at a brain region that I'm highlighting here, and it's part of the thalamus. The optic tracts continue to the thalamus. Then from the thalamus, we get more axons. So these are separate neurons. We get more axons that pass to the visual cortex. The term for the axons that are passing in this part, these are called optic radiations. So we have three terms for different groups of axons. Optic nerve going to the optic chiasm, optic tract going to the thalamus, optic radiations going to the visual cortex. If I were to draw every single neuron in this pathway, if you recall, for vision, we use photoreceptors. So if I'm going to do an inset here, of the retina. In the retina, we have photoreceptors. I'm going to color that in green. So there's a photoreceptor. And then vision is a special sense. So the next cell in line is a bipolar cell. And it's literally called a bipolar cell, bipolar neuron. The next neuron in the pathway is another bipolar neuron. It has a unique name. It's called, whoops. It's called a ganglion cell. So we have a we have a photoreceptor, that's neuroepithelium. We have a bipolar cell, that's a neuron. We have a ganglion cell, we have another neuron. And this neuron exits the retina as the optic nerve. It either does cross or doesn't cross at the chiasm. In this case, we're not going to cross because it's coming from our, it's coming from our right visual field. And then this neuron ends at the thalamus. So the ganglion cell starts at the retina, optic tract, optic chiasm, optic nerve, optic tract. One more time. From the ganglion cell starts in the ganglion cell starts in the retina. Ganglion cell starts in the retina. Exits the optic nerve. Does not, in this case, cross over at the optic chiasm. Goes through the optic tract, ends at the thalamus. There we go. From the thalamus, we get another neuron. I'm going to draw this neuron in black. This next neuron, it's multipolar interneuron. It's going to go through these optic radiations and at the visual cortex. That's how basic vision is processed. From the retina, photoreceptor, bipolar cell, ganglion cell, to the thalamus, where we get another neuron. This is a multipolar interneuron. Those are the first two cranial nerves. To understand the next cranial nerve, and actually the next few cranial nerves, we need to first understand the muscles that move the eye. The muscles that move your eye, so to look left, look right, look up, down, to even rotate your eye, these are called extraocular muscles. Extra because they're outside of the eye. Ocular, it's moving the eye, outside of the eye, extraocular. There are 6 muscles that control movements of the eye. So I have pictured here in the middle, a picture of a right eye. So if this is the right eye, that means your nose would be right here. That triangle is supposed to be a nose. If you want to look towards your nose, let me draw the nose again. If you want to look towards your nose, you need to use your medial rectus. Medial rectus, look towards your nose. So that would be adduction. If you want to look away from your nose, you'd use this muscle, the lateral rectus. If it wasn't apparent, these are called medial and lateral rectus because they're more medial, they're more lateral, and rectus means straight. These are straight muscles, thin, straight muscles that pull in one or the other direction. Medial rectus for adduction, lateral rectus for abduction. Superior rectus for elevation, looking up. Inferior rectus for depression, looking down. We have 2 more muscles and they do something a little unique. and they're unique in how they're attached. Take a look in this yellow marker. I'm going to draw a muscle in the middle. See, the muscle is right here. It comes through this little loop and then attaches at an angle. So it's hiding behind the superior rectus a little bit, but it attaches sideways on, along this transverse plane on the eye. This is called the superior oblique. So because it goes through that little loop, through that little pulley, if you pull on it, it's going to make the eye rotate a bit. It's going to rotate the top of the eye towards the nose. So in effect, it's just making the eye look down and in, but it gets there by rotating. The term for that is intorsion. The superior oblique muscle is for intorsion. The other muscle, if you look in the middle picture over here in red, comes also at an angle. It doesn't go through a little hook. It doesn't go through a little pulley, but it attaches at a side transverse plane. This is the inferior oblique. And if you pull the inferior oblique, it's going to make the eye move up and in. That's extorsion. 6 different muscles that control eye movements. Given that, we'll be talking about 3 different nerves, 2 in the coming videos, actually 1 more in the coming video, 3 cranial nerves that control eye movement. Here's the first one. The oculomotor nerve, cranial nerve 3, is a motor control nerve. So it's not sensory, it's motor. And it controls the superior rectus, the inferior rectus, the medial rectus, and the inferior oblique. It controls 4 of those 6 muscles. Since this is a motor nerve, that means you have a neuron starting somewhere in the brain. It's going to start in the midbrain near the superior colliculus. If you look down here, this drawing of the brain, let's pretend we have a neuron cell body here in the midbrain. I'm going to draw it as a multipolar neuron. Its axon extends out of the midbrain. And let's have it go to the superior rectus muscle. So there's one neuron starting in the midbrain, exiting and controlling the superior rectus. I could have this go to any of the other muscles that I've mentioned here, but there's one example. This nerve passes through a hole in the skull. The hole that it passes through is a superior orbital fissure. It's that long crack at the back of the orbit. It's part of the sphenoid bone. Right, so when that doctor does that eye test and have you follow their finger, one of the nerves that they're testing is the oculomotor nerve. There's other things that it's important for besides moving these 4 muscles. Take a look at the bottom left. Another muscle that you can control is the muscle that lifts your eyelid. The muscle that lifts your eyelid is above, it's sitting on top of the superior rectus. It's not attached to the eye, it's attached to the eyelid. You can see it right here. This is the levator palpebrae superioris. Levator means to lift. Palpebrae means eyelid. Superioris means your upper eyelid. Or superioris means upper. Levator palpebrae superioris. Raise your upper eyelid. Your oculomotor nerve controls that too. If you want to focus on something in front of you, like right now I'm focused on you, and then I can change my focus. If you look at my eyes, now I'm focusing on this pen. To do that, that requires muscles. Those muscles are found in something called the ciliary body. And the ciliary body is found deeper in the eye, like over here. More like, that's not actually representative. More found over on these edges over here. There we go. It's posterior to the lens, or actually it's surrounding the lens. That's a better way to say it. It's surrounding the lens. If you don't know what those parts are, we'll get to them. But there's this area within the eye that controls the shape of your lens, which helps you focus. They're called ciliary muscles. I know you've heard that word cilia before. Cilia just means hair, and these muscles look hair-like, and they have little hair pull extensions. These ciliary muscles are controlled by oculomotor nerve 3, and they control how you focus. The term for focusing on something, for adjusting your focus, that's called accommodation. You're changing the shape of your lens. Cranial nerve 3, oculomotor nerve, controls ciliary muscles, allows you to focus on something. One more thing that we can control with this nerve, and we can control the muscles that surround your pupil. Your pupil is the hole that lets light into your eye. You can make that pupil bigger, you can make the pupil smaller. You can dilate, make it bigger, you can constrict, make smaller. To change your pupil constriction, to make it smaller, that's the sphincter pupillae muscle. It's found in the colored part of your eye, found in the iris. The colored part of your eye is the iris. Iris does more than just look cool and have color and absorb light. It actually has muscle in it that can change the size of your pupil. If you know that test where you shine a light on your eye, if you shine a light on your eye, the pupil should get smaller. That's controlled by oculomotor nerve 3. So a lot of things controlled by this one nerve. It's not that big, but it's important for all these things relating to the eye. Not all muscles of the eye are controlled by this nerve, just some of the muscles. And it does not have anything to do with vision. It only controls movement. It only controls these muscles. I mentioned the midbrain. We've only talked about it a little bit before. What is the midbrain important for? If we look at, first let's orient ourselves. If you look at this picture up over here, we've taken the brainstem and the thalamus out of the brain, and now we're looking from a posterior dorsal view. So you can see the thalamus here. That means the third ventricle would be here in the middle. You can see the pineal gland back here. The midbrain is this region. The pons would be more caudal. The pons would be over here, and then you get the medulla. We'll get to those later. But the midbrain is in between the pons and the thalamus. And generally, the midbrain helps you maintain alertness, helps you maintain consciousness, help you just be alive and awake. Sometimes when people fall into comas, they have damage to this midbrain, because if you're in a coma, you're not really conscious, or at least not alert. You might have some activity going on, but you're not alert and awake. There are these two regions of the midbrain on the dorsal posterior side. Do you see these bumps? There's one set of bumps there in pink, and then there's another set of bumps here in dark blue. The top set of bumps are the superior colliculi, singular superior colliculus. The bottom set of bumps are inferior colliculi, plural or singular inferior colliculus. Together, they're known as the corpora quadrigemina, the four twins, or four quadruplets, rather. The superior colliculi in particular, what are they important for? Why am I bringing it up now? The superior colliculi are important for mediating a visual startle reflex. If you see a bright flash of light, what you'll automatically do is flinch. And flinching can involve, like getting tense with your shoulders, or closing your eyes, or moving your head. It's flinching. It's completely normal. It's reflexive because if there's a bright flash of light, that might mean there's something coming that might hurt you. And so you want to protect your eyes, protect other things. That's mediated in part by your, not only by seeing it, so your optic nerve, but also controlling what your eye does, your oculomotor nerve. And in between, you're sending information to this brain region. You're sending information to the superior colliculus to control that, to regulate that information. You're getting information in, deciding what to do with it, sending output to control something. One more nerve before we switch videos. We'll split up 4 by 4 by 4. The trochlear nerves. Trochlear nerve is cranial nerve 4. Trochlear nerves are also motor. So we've gone sensory for olfactory, sensory for optic, motor for oculomotor, motor here for trochlear. The trochlear nerve controls only one muscle, or I guess one pair of muscles. It controls the superior oblique muscle. Remember this one that's hiding and goes through this weird little pulley? It controls the superior oblique. It's called the trochlear nerve because that muscle goes through a little pulley. The term for pulley is trochlea. So you have a little trochlea here where the tendon of this muscle passes through. The trochlea is made of cartilage that's hanging off of your maxillary bone. Or actually, sorry, your frontal bone. It's hanging off your frontal bone. You can see it in this picture down here. It's the smallest cranial nerve because you're only controlling this tiny muscle on either side. You start in the midbrain. So I'm going to draw a multipolar neuron here, multipolar interneuron here. Its axon is going to exit out of the superior orbital fissure. If you recall, that's part of the sphenoid. And then terminate at the superior oblique muscle. All it does is control the superior oblique eye movement for intorsion. Controls intorsion of your eye. All right. Those are cranial nerves 1 through 4. In the next videos, we'll go through 5 through 8 and then 9 through 12. So I'll see you in the next videos and we'll practice more in looking at this with an actual brain and some 3D models. I'll see you all later. Let me know if you have any questions.